All right, hi everybody. Today we have a new micro seminar. Despite the funk that I've been in this morning, given proposed budgets and whatnot, but we got a new seminar today, so this will hopefully cheer everybody up. Also, I need to pump Lenny's Protocols IO. He gave me a T-shirt, so he told me that I needed to. He gave me a T-shirt for other reasons, but we are doing this on a strictly voluntary basis. But I owed him showing the Protocols IO stuff to people. All right, so today I'm actually pretty excited because we are starting into the first of the scientists on I think maybe Tom was the first. This is the first from the U of A campus besides me. So today we've got another fellow researcher here at the University of Arizona. I apologize if there's any timing links and stuff that were screwed up because we in Arizona are the only smart states that don't have daylight savings time. So I kind of screwed that up myself internally. But today's micro seminar is from Dr. Michael Johnson. Michael's got a pretty diverse background. It's interesting in a few different ways. First thing, he started off at Duke as a music major. I have a daughter too. That's asking me things right now. So Michael started off as a music major at Duke a while ago, which has led him to weird allegiances because he went to Duke and then started to. After going to Duke as an undergrad, went down the road to be a technician at the better university in North Carolina, UNC, and then stayed at the better University in North Carolina for a few years after that, both as a tech and then to get his PhD in Jeff Frellinger's lab, or Matt Redenbaugh's lab. So he was a tech for Jeff Frellinger and then got his PhD in Matt Redenbaugh's lab. After his time in beautiful Chapel Hill and after surviving his time in Duke, Michael then went off to do a postdoc in Tennessee at St. Jude's, where he started some of the work that he'll talk about today. So he was a postdoc for 3 years in Jason Roche's lab, and then went to work with Doug Green for a year and a half. And then lastly, started off here at the University of Arizona in beautiful Tucson, about almost a year, July of last year, July of 2016. So I think Michael's going to talk about copper today. He's going to talk about probably Pseudomonas and give you guys a pretty cool view into how pathogens deal with heavy metals. So without further ado, Michael Johnson.
All right. Hi, everybody. My name is Michael Johnson. Let me get this talk started for you. There we go. And today I'd like to tell you about how copper is toxic and why we should care. So one of the first things I want to do, just in case you're coming back to this talk later and it's archived, is this is the too long didn't read slide. So you can kind of come back and reference these things, but hopefully by the end of this talk, everything on this slide will actually make sense to you. I'll actually start off by asking a question. Do you all take vitamins? So you, sir, with the MacBook Pro with your coffee next to it, I'm wondering, no, I'm kidding, I can't see you. Hopefully you laughed. I can't see you right now. But just wondering, so when you look at your vitamins at the end, you actually see a lot of metals. So you can see kind of in this area, you see calcium, iron, magnesium, zinc. I'm actually interested in this one, copper, which the daily recommended value is about 2 milligrams. So what happens when bacteria enter your body? So when bacteria enter your body, they need certain things. They need these metals. So metals account for about 40% of metals are in about 40% of your proteins. They account for like structural components or enzymatic factors. So 40% of your proteins don't work. Of course, you don't work. So bacteria, they can't bring suitcases in with them. So they have to acquire all of those things from within you. The host knows this and actually tries to reuptake or chelate some of these metals. This happens inside the phagolysosome of the macrophage. So it tries to reuptake some of these metals, but it also tries to pour in some of these toxic metals like copper and zinc. So, and that's bad for the bacteria, and the bacteria knows that. So it actually tries to now efflux some of those metals like copper, zinc, calcium, and manganese. So you'll see calcium and manganese in both sides. That's because too much of it is actually a bad thing. Interesting to note, some of the bacteria have a lot of iron import systems, but no known iron export systems. And they have copper export systems, but no known copper import systems. So that's kind of a testament to how necessary iron is and how much bacteria don't like copper. So we're going to focus on copper today, which is actually universally toxic to pathogenic bacteria. So a brief history of copper. So it was used as kind of copper pots to help store food. You know, the food wouldn't spoil as quickly. They had no idea why. They just knew that it happened. People actually sprayed things with copper onto some grapevines. It was actually known as the Bordeaux mixture because you got to have your wine, right? Same mixture actually helped prevent potato blight. And then you kind of keep zooming into today, and it actually can cut down on cross-contamination inside incubators. Sometimes you toss this copper disc inside of the water and in your incubator, and that keeps it actually cuts down on some of the bacterial growth. Or actually, inside of hospitals, one of the things you can do is you can actually use copper tools or copper surfaces, and it's been shown to decrease nosocomial infection over stainless steel. So bacteria have a universally conserved export system. So I'm going to kind of walk you through that. The nomenclature here is specific to the bacteria I'll talk to later, which is Streptococcus pneumoniae. However, all of these players exist in bacteria. So you have this COP-Y, which is the repressor, which binds upstream, kind of like the chaperone sees copper, opens up. So there you have it. It binds copper, opens up, makes more COP-Y, which is the repressor. I would have called that COP-R for repressor, but that's just me. Then you have this COP-A protein, which is a copper chaperone, which I would have called COP-C for chaperone. And this chaperone's sole responsibility is to take copper to COP-A, which is the export, which I would have called COP-E. So again, sorry about the nomenclature, but what are you going to do? So COP-A takes copper to COP-A, and COP-A has been known to export copper-1. So the point of this talk, so the observation is bacteria have evolved with copper present, yet copper is actually still toxic to these things. And we have very little understanding of how this happens. What are the mechanisms of toxicity? What are the bacteria doing to overcome some of this stress? So we really want to examine the orchestrated response, kind of everything that the bacteria is doing in response to this copper. Hopefully we can use this to circumvent some antibiotic resistance, because bacteria have adapted to antibiotics very quickly. And like I said, copper has been toxic since antiquity and still is today. Hopefully we can come up with some kind of synergistic molecule to with copper or with the actual host to prevent some of these bad infections or keep them at bay. But to do that, first we need to actually understand what are the major players in these particular systems. Okay, so the bacteria I'm working with is Streptococcus pneumoniae. Streptococcus pneumoniae, or pneumococcus, causes a lot of bad infections. You have meningitis, you have ear infections, bacteremia, and of course, pneumonia. It can go into the bloodstream quite easily. Unfortunately, it's carried asymptomatically by many people, so it can actually be passed on. There's over 90 different serotypes, which makes battling this bacteria kind of difficult. Even though we do have a vaccine, it only covers maybe about a quarter to less than that of the actual serotypes of Streptococcus pneumoniae. And those are often characterized by the outer shell or the capsule layer of these particular bacteria. And unfortunately, over 1 million deaths are associated with Streptococcus pneumoniae per year. Many of those are children under the age of 5, just to pneumonia alone. So we want to first look at the members of this particular COP operon inside of Streptococcus pneumoniae. We want to know that we have the right system to look at and knock out those proteins and see exactly what they're doing. So when we knock out when we look at the regulation of COP-A in the presence of copper, we see a nice increase that is copper dependent. This is the increase of copper. The other thing we can do is we can do an endpoint growth curve. So basically we have the bacteria, We start them at a certain OD. We back dilute them in a media with a certain amount of copper, 50, 100, 250, and so on and so forth. And we see what their growth is the next day. And you can see there's a nice dose-dependent response to the wild type, which is Tiger 4. When you knock out the repressor, you're making lots of the chaperone and exporter, so that's not really going to have be susceptible to copper stress. However, if you knock out the chaperone and you knock out that exporter, now you have a almost a tenfold increase in susceptibility to copper stress. So that's in vitro, but does it matter in vivo? So we can now take these bacteria and look at them in mice. So you look at the COP-A mutant or that exporter mutant, and there's 100% survival of these mice. This is a pneumonia infection, so intertracheal. We've also done IV, we've done IP, and very similar results for each one of those for this particular situation. We can also look at the lung titers, and we can look at the blood titers, and we can see significant decrease among those particular situations. So there's actually another Streptococcus that we deal with, and that's probably the Streptococcus people here know about more because they get strep throat, and that's Streptococcus pyogenes, but it can also cause a couple other things like Streptococcus, sorry, I'm getting ahead of myself. One more slide about the COP-A virulence inside of Streptococcus pneumoniae. Here's actual otitis media or ear infection, and then here's a sinusitis model. So you can see a drastic reduction in the incidence of in the presence of having that mutation. So that's keeping the copper inside of the bacteria. So here's what I was talking about. So this is actually a mouse model of infection for Streptococcus pyogenes. This is actually a necrotizing fasciitis, where you can just induce a wound on the skin, and you can look at lesion size with the wild type, which is this HSC5 and COP-A, and the bacteria, per gram of the lesion. You can see that's also reduced in the COP-A mutant. So present is leading to less virulence overall. So another interesting thing we can do is we can now look in the, again, look in the mouse model, and we can look at those cells that we thought were, characteristic, or that were engulfing some of those bacteria, having them in the phagolysosome to actually clear the bacteria in a copper-dependent manner, as I showed the nutritional immunity earlier. So when you look here, what we did was we used clondronate liposomes, which basically deplete macrophages in the area. So we added it to the lung, and we could deplete the lung macrophages. And you can see there's no significance here. However, about maybe 2 logs here in the mock infected. And if you recall the last lung slide, there was also a big difference there between the Tiger 4 and the COP-A. Additionally, what's also interesting is, even though we knocked out the macrophages in the lung blood, we knocked. So when you do that, you actually see you see that decrease in the blood. So what this is telling us is that the bacteria aren't getting through the lung into the bloodstream that are basically susceptible to copper stress. So that copper is actually causing a lot of things to go on inside of that bacteria, some of which I'll get to in this talk. So the takeaway message from here is copper-mediated toxicity from macrophages strongly affects Streptococcus pneumonia survival during infection. So your body is already providing the copper that's needed to kind of combat these bacteria. Your body is very good at regulating metals. Too much copper is bad, too little copper is bad. There's that Goldilocks zone of metals for each one. So giving more metal is not necessarily the answer, but we can come up with potential small molecule therapeutics to target some of the other pathways in the bacteria. So again, that's where we're going to try and go with this. So I mentioned they don't make it through the lung to the bloodstream, so that might have something to do with adhesion. So we looked at some lung epithelial cells, saw that there was a defect in adhesion with the COP-A mutant, and that actually corresponded to even more drastic decrease in invasion. So interesting to note, there's something going on with the copper inside that's leading to a decrease in adhesion and decrease to invasion. which there's some other genes that may be responsible for adhesion and invasion that are turned down. We're currently investigating some of these things, but we have no idea how copper would be regulating them. So again, that's something that we're really interested in going into. So again, it enters the epithelial cells before being caught by macrophages. So it could be that they're replicating faster. It could be a lot of variables that lead to that lack of adhesion and lack of invasion as compared to wild type. So right here, we're rebuilding this model from the beginning. We're using copper-2 in these assays because that's actually the soluble form. Copper-1 is insoluble for the most part, but you can try and get it soluble under reducing conditions, but it's kind of tricky. So we have this COP-Y, sees copper, opens up, and you start making more of these particular operons. And when you block COP-A or delete it, that decreases virulence, decreases attachment, decreases invasion, and decreases the resistance to copper stress. So 2 theories on copper toxicity. One is just oxidative stress. So you can have basically copper-1 combining with hydrogen peroxide, and that basically leads to some of these oxidative stress, bad things that will tear up your DNA and tear up your proteins. Thing about that is Streptococcus pneumoniae actually makes lots of hydrogen peroxide as well. So that is actually a theory that could hold water. Another option is mismetallation. So that could be some of those enzymes that are using metals for turnover. It could actually be locking those up, or competition for import could lead to protein misfolding, or even worse, protein unfolding. So we wanted to look at oxidative stress first. So this is basically a oxyblotter looking for oxidized proteins under copper stress. So we can increase the amounts of copper, and we look at Tiger 4, we look at COP-A, and lo and behold, we see that there's actually more oxidative stress inside of our COP-A mutant. However, when you look at some of the other mutants, if you recall, the COP-A mutant is actually copper sensitive, but that has less oxidative stress than the wild type. And if you look at COP-Y, which if you recall had wild type susceptibility to copper stress, we have a lot more oxidation here. So oxidation, it may play a small role, but according to this, it is not one of the ultimate mechanisms of copper toxicity inside of Streptococcus pneumoniae. So we had to keep going. So one of the things we did is we wanted to know if we could rescue copper toxicity. Is there a battle for import? In one of the importers, we looked at ICP mass spec, which is basically looking at the internal metal concentrations under various conditions, and we didn't see any upregulation or downregulation of metals inside the bacteria under copper stress. So it wasn't necessarily a competition for import, but we did see that manganese actually rescued copper toxicity. So you give them, a dose of copper, you compare that to the wild type, no copper growth, and you can actually see an increase in manganese. That it actually increases the growth by about was that maybe 33% there. And you can also look at some other ways. You can see manganese rescues bacterial growth. See the CFU increase in wild type and COP-A. And you can also see that when you try and do macrophage killing, you basically add the bacteria to macrophages and see how long they survive. The manganese actually can help rescue not only the wild type, but also the copper mutant. So we have a microarray of some of these things. And these are some of the things I'll talk about later on in this talk, but I want to introduce them here. So right here and highlighted in red, we basically had this anaerobic nucleotide synthesis pathway in Streptococcus pneumoniae. Now, strep pneumo can grow anaerobically and aerobically, and it has dedicated nucleotide synthesis pathways for both processes. So when you look at those, we saw that they were turned up here, and we look at some of the anaerobic and aerobic, they were all turned up. So when you look at these particular pathways, you see Here's the aerobic side, and also here's the anaerobic side. You see that this this NERD-F protein is actually manganese dependent. So we were able to rescue with manganese, and this particular catalytic enzyme in this pathway is manganese dependent. So the other thing to note is that these pathways can actually complement each other, and that if you try and knock out some of these aerobic genes, they're essential unless you grow them all in anaerobic conditions, which we did not get to. So we did the next best thing. We knocked out NERD-D, which is the one of the crucial proteins in the anaerobic system. So what does that NERD-F protein look like with the two manganese ions? So you can see in green is the actual crystal structure of NERD-F. You can see these two manganese ions bound here. Closer view is how they coordinate here. And when I modeled the Streptococcus pneumoniae NERD-F, it models very closely to that E. coli structure. Interestingly to note that these particular binding residues are also very prevalent in binding copper-2. Not copper-1, but copper-2. Copper-1, the binding motifs are that CXXC or CXC motif, which should sound pretty similar if you are. You would probably recognize that if you're in the immunology field, some of those CXC motif chemokines. So up here is actually the Urban-Williams series. The Urban-Williams series defines how stable a metal is in an active site. So inside of that active site, you have manganese, which is kind of unstable, which means you have on, off, on, off, on, off. Versus copper, which is more stable, would go in there and basically just clamp it down. At least that was our hypothesis. So we tried to, since we didn't mutate the aerobic system, we did the next best thing, which we chemically inhibited using hydroxyurea. So here's that same kind of growth curve I did earlier. You can see there's a certain amount of a fraction of growth when we add hydroxyurea. I believe this was 10 millimolar. Now, when we added it to the NERD-D mutant, so now we're inhibiting both the aerobic pathway using hydroxyurea and the anaerobic pathway because of the mutation, and we see zero growth. Now, if the if copper were actually playing a role in this particular system, then we would expect to see a significant decrease. But if copper was not playing a role, we would expect to see to be more like wild type. And we do see that decrease as compared to the wild type, implying that copper is indeed involved in the aerobic nucleotide synthesis pathway and poisoning that pathway. So again, here's another model. You have nucleotide synthesis, NERD-F making lots of nucleotides. Copper comes in, replaces those manganese ions, and now you have reduced nucleotide pool. And if you can't make nucleotides, you can't make DNA, you can't replicate, and you're kind of a sitting duck for the immune system. So this is why the bacteria tries to upregulate the alternative nucleotide pathway, or the anaerobic. I didn't mean to have alternative in there. Sorry about that. No jokes. Okay, whatever. Okay, so here's an interesting proof of concept and proof of other targets of copper toxicity. So we looked at hydroxyurea and Streptococcus pyogenes, and we saw there was no significant difference between the wild type and the copper A mutant. So when we did some other digging, what we found out is that Streptococcus pyogenes actually has a manganese-independent pathway, nucleotide synthesis pathway. Copper is still toxic to that particular bacteria. We can, make those mutations to COP-A, and that's susceptible to copper more than the wild type, as with Streptococcus pneumoniae. And I showed you the virulence data as well. But it has a completely different mechanism of toxicity, which we still don't know. So, again, this is kind of a proof of concept of not only some of the for these toxicity mechanisms, but also proof that there are other targets as well. So now what do we know? So, again, we have this operon, sees copper, releases, makes more COP-A to export copper. We know that now copper can block nucleotide synthesis, but manganese can basically rescue that phenotype. We also know that the anaerobic nucleotide synthesis pathway is turned up under this under copper stress to try and overcome this. So one of the other things that was on the bottom of that little microarray chart was also that zinc, or this CZCD, which is a zinc exporter, is also turned up. So insert the brass dagger. So, I was I mentioned I was a music major at Duke. Also, yes, I still root for Duke all the time, and we did beat UNC 2-1 this year. Maybe we'll see you in the national championship and we can make it 3-1. I know you're on mute and you can't talk smack to me back, so haha. Anyway, so when we look at this same rescue, we look at the combination of copper and zinc, which of course makes brass, and that actually has a decrease in growth for the particular bacteria. So playing a brass or playing a trumpet, I kind of put this together later in life. We're blowing hot air into this trumpet, stuff from our mouth, which is kind of gross when you think about it too much. Then we take, from this warm trumpet and put it inside of a nice incubated case, and then we zip that up. And I mean, that should be a complete petri dish of absolute nastiness. But what's interesting is it's made of brass, which is copper and zinc. So that's why you actually, don't get a lot of that, bacterial buildup inside of that trumpet because of the outside surface is actually abiotic. But that doesn't address what exactly now is the zinc doing. So we decided to purify some of the proteins involved in that copper export pathway. And lo and behold, when we look at the repressor's ability to bind the DNA upstream, we see that by adding zinc, we increase the affinity for the DNA about 10-fold. Now, what's really interesting with this is, sorry, am I still on, or?
Yeah, you're good. You're good.
Okay, I saw the green light go off. All right, let me turn that back. Come on, there we go. All right, so we see a 10-fold increase in the affinity for DNA with zinc present. So for these experiments, we purified the COP-Y protein. We ran it over a Chelex resin to chelate all the copper or all the whatever metal might be present. And then we concentrated that and used that for our experiments. That way we could actually add back more zinc. So one interesting thing with this is we also did circular dichroism, which is basically a way of looking at the secondary structure of a protein. Does it have more alpha helical signal, or does it have more beta sheet signal? And when proteins are more in the DNA binding conformation, they have more alpha helical signal. So when we add zinc, that actually looks more like the apo form, versus when we add copper, it actually decreases some of that alpha helical signal. So we decided to look a little bit deeper at some of these copper export mutants. So we made So we made the zinc exporter mutants, and of course they're susceptible to zinc stress. We see some differences here between the COP-A and the CZCD COP-A mutant here. But when we looked at the wild type and the CZCD, we didn't see an immediate impact until we looked at the kind of the time-based study, and we could see that there was indeed a lag. So again, this is zinc competing with copper to try and open up this operon. So there's maybe a 2-hour lag in the ability of the bacteria to grow. And when you so that's and then here's kind of too much copper here's just basically the regular growth, and then in the star you can see that that's the lag as compared to the wild type under the same condition, which is the upside down blue triangles. So there is this lag. So zinc actually is inhibiting this particular operon from opening up, and that does have detrimental effects on the bacteria under copper stress. Oh, there we go. Another interesting thing that we did was we wanted to know what zinc did to the protein as far as multimerizing it, because we saw some interesting shifts on a gel when we ran the COP-Y protein with DNA via some EMSAs. So we saw this kind of super shift when we added zinc. It turns out that what ends up happening is when you look at the apo form as compared to the zinc bound form, it goes from a dimer. So the molecular weight is about 18. So when there's no zinc present there, it's a dimer. So that's about 40 kilodaltons, versus when there is zinc present, you actually see a duplicate. It basically doubles that to 75. So it actually makes it a tetramer. So now we can further enhance our pathway. We can see that, okay, now there's a tetramer that's bound upstream of this operon here. And we know that once copper leaves the bacteria, remember, it actually has a zinc import system, so it can still actively import zinc. So to close the system, you actually can have zinc binding to copper-Y to return back to the operon. So it can basically, zinc actually helps close the system. So now I want to focus on the chaperone protein and what I think that's doing. So it turns out that it has this transmembrane domain, which helps it traffic to COP-A in the membrane, which makes sense. Has this copper binding domain down here, which is how it does the pass off. But it also has this cuprodoxin domain, or something that helps reduce copper. So why would we want to reduce copper? So as I mentioned, copper-1 is pretty insoluble, and we've been doing all of these studies with copper-2. So copper-2 enters and copper-1 leaves. So you need a source of electrons, or you need to be able to reduce that copper for it to actually be exported in the bacteria, exported from the bacteria rather, sorry. So couple questions. How exactly does that chaperone acquire copper? Can the chaperone protein facilitate the reduction of copper as only copper-1 is exported? And where could those electrons be coming from? So these are all potential places we're building up our repertoire of where we could potentially target with a small molecule therapeutic. We really have to understand the system before we can start to manipulate it. Or at least we should understand it before we start to manipulate it. So Dave Gierock's group published a really nice structure of CUP-A, which is a chaperone protein. It has two major sites, this kind of site 1 here between these two cysteines, and this site 2 here, which actually helps pass the copper off to COP-A with these methionines here. So our model was copper-2 can come in, it can actually be reduced on contact from these cysteine residues, and then it's passed over to the methionines and exported. But if these cysteines actually reduce this copper, they would need an extra electron to be able to grab copper-2 again. They could grab copper-1 because of that binding moiety, but to actually grab copper-2 to reduce it on contact, they need another source of electrons. But where is that copper-2 coming from? Where's the copper-1? Is it just free floating in the bacteria, inside the bacteria, or could it actually steal it from COP-Y? So we decided to test this one first. So I mentioned that we have both of these. We had the COP-Y repressor. We also made the CUP-A chaperone protein. So what we did was we bound COP-Y to an affinity column with nickel. We had we added copper and washed it so that only COP-Y was bound to copper with no other copper on the column by washing it. We flowed over TEV cleaved CUP-A, so it could no longer bind to the column via his tag. And then we looked at the amount of copper basically bound to CUP-A as it was eluted via ICP mass spec. And we can see it was drastically increased compared to the COP-Y column that had no COP-Y or no copper present. And we've actually done some EMSAs to characterize this as well and to confirm this data. So you can run an EMSA, which is just an electromobility shift assays, DNA binding. You look for the shift up if it's binding protein. If it doesn't bind protein, then the DNA runs faster. So when you give copper to that particular mixture of COP-Y and DNA, you see that the DNA runs all the way to the bottom because the protein can't bind. If you add CUP-A to that, it can now chelate the copper as it does here, and now it actually restores binding of the repressor to the piece of DNA. So now we want to see, okay, can copper reduce? Can copper be reduced by this chaperone protein? So we have this bathocuprine disulfonic acid. Say it 3 times fast. And it actually is a reporter for copper-1. It absorbs at 490, and we can detect that in a variety of different ways in a variety of different systems. So if we take this BC compound and we add copper-1, we see it turn orange. If we add copper-2, it can use potential electrons from somewhere, but it's a much slower process. This one happens in literally milliseconds versus this one happens in maybe 3 days, and that's more from maybe the oxygen from the environment that's actually onto the plate. But for the most part, it does not give any signal, and if it is, it's very, very minimal. So what would happen if we had this BC and we added copper-2 and CUP-A? And what can we check for? We can check for top reaction rate, how fast these reactions can occur. So first things first, we wanted to see, does CUP-A, which is the wild type, actually give us a signal? So we increase the amount of copper, and we see this nice Vmax with CUP-A, very nice, and a Km here of 9.962. So we can actually see a curve. We see that it is actually reducing copper-2 because that's where we're adding here. So we can basically add copper-2, it can be reduced, and then it gets passed off to that BC compound. What happens when we mutate some of these residues? So if we mutate this cysteine or this cysteine, it should drastically slow down the overall rate, and that's exactly what we see. We see a drastic reduction, and if we mutate both of these, we see some non-specific binding there. Interestingly, with this system and testing it, interesting with this system, which actually really, I think, validates it quite nicely, is when we mutate some of these methionines, we now see an increase in that rate. So I'm going to use a football analogy here, actually. So imagine you're a quarterback and you're the quarterback and you're handing off, the football is actually copper-2. Okay, so if you throw it to the running back and the running back has 2 hands, it can catch the ball very nicely, and bang. If you tie one of those hands behind the back, maybe, or a wide receiver, maybe if you have Odell Beckham present, you can still catch it and still potentially reduce it and hand it off and facilitate that reduction. However, if you actually reduce the copper via these sites, and you're old school Tiki Barber and you can't hold on to the ball, you actually increase the amount that the reporter can actually bind to that copper. So that's why you see that increase of rate here. It's not an increase in the reduction, it's just an increase in the rate as detected by the reporter system that we have. Additionally, we can make mutations to this cysteine, methionine, or double mutant. Each one of these actually makes the bacteria more susceptible to copper stress as compared to the wild type. So we knocked out the CUP-A protein, and we added back plasmids with each of the mutations. So here's our revised model. So copper-2 can come in, copper can be reduced, it gets passed off to this methionine residue, and then it goes to COP-A. We know that it can come from COP-Y. Now, interestingly, the previous dogma was that the COP-Y repressor gets the protein from CUP-A, the chaperone, which I didn't necessarily understand, because how do you have chaperone present if the repressor is still bound? I guess some operons can be leaky, but I think this version of it offers a much more robust system that you can basically open up by the repressor seeing copper and then closing it via zinc. Again, and also as a potential source for the electrons, we see this thyrodoxin is actually turned up under copper stress. So that could actually be responsible for reducing CUP-A. And there actually is literature to support that thyrodoxin reduces copper reducing proteins or at least proteins that facilitate copper reduction. So now we kind of are building on our model even more. We have our initial toxicity. We have our homotetramer here, copper 1 coming out here, zinc kind of closing up this whole system. But we have COP-Y here with copper 1 or potentially copper 2 bound. It's kind of hard to tell which one it is because we have no idea how COP-Y actually interacts with copper 1 or copper 2 or zinc for that matter. We just know that they do. So that's some structural characterizations that we're currently in the process of doing. So in doing so, the cup A can actually chelate these this copper from COP-Y, or just get it free into the from the inside of the bacteria, and then copper reduction can occur and then export. So you have this interesting conundrum, like, oh, inside the bacteria is a reducing environment, but it's to what degree is that environment reducing enough to facilitate all of this incoming copper? So, I mean, the electrons have to come from somewhere. So, you know, at what is the line between being a reducing environment and an oxidizing environment versus, in, you know, some of these little micro pockets of the bacteria. So it's probably not a homogeneous environment. There are some of these pockets that can actually facilitate this copper 2, as I showed that it bound to that NERD-F protein, and I showed that this protein can actually actively facilitate that reduction. So I mentioned we had a microarray. So what are some of the other systems that were altered? So we saw that thyroidoxin system. We saw some other ABC transporters, competent stimulating peptides. So that's responsible for trying to have gene transfer from other bacteria, trying to, you know, like almost on SOS, we need more genes to maybe overcome some of the stress. Not an SOS response in the traditional sense, but like just trying to get excess DNA to try and find any way it can be resistant via homologous recombination. We also have these clip proteases that were turned up that can actually degrade protein and have been shown to be involved in virulence. We have some other antiporters, some other regulators that could be responsible for some of the gene regulation, responsible for looking at or responsible for mitigating some of that copper stress. And there's actually quite a few unknowns as well in some histidine kinase sensors. So these are all things that we're actively trying to pursue to really tease out what the mechanisms of toxicity, what some of those other mechanisms are, and also what the bacteria is doing to overcome some of these this copper toxicity. Because hopefully, if we can block what's being used to overcome copper. If we can block what's being used to overcome some of the copper stress, then we can again synergize with the host inside of that macrophage to eliminate the bacteria faster. Long-term goals. So we really want to see, perhaps bacteria can use this metal sensing or metal, its metal environment as a means to determine location. I showed earlier that it showed that that under copper stress, it was less adherent and less invasive. Is that maybe a mechanism to escape the environment that it's in? Oh no, I see copper, and if I see copper, I need to turn up all my resistance and virulence pathways and try and get away from whatever's trying to get me. But can we also take some of the things that we learned here and look in other bacteria and potentially expand that to other toxic metals? Quite interesting, other metals that open up that copper operon via COP-Y is actually gold and silver. So I guess that protein has very expensive taste, but those aren't necessarily biologically relevant, but it does kind of open the door to other metals, and people have used silver in the past. And we also want to see, can we now use this information to see if what are some of the other metal-specific things that the host is doing that combat infection? Because now that if we know what the bacterial response is, perhaps we could extrapolate what actually is what it's responding to from the host. So I'd like to thank some of the people in the lab that's done some of this work. So we have Miranda, who's my lab manager, an undergrad Michael Trial, a few master students in Preetham and Itzel, and the lab where I started this work, Jason Rosh's lab, and a very nice undergrad who's now MD PhD at UT Memphis. And I'd like to also thank the department, Bio5 here for funding. I'd like to add R1 up there. Hopefully, I'll be able to add more than 80% of one, and also the College of Medicine. And with that, I can take any questions you have.
Cool. Thanks much, man. I will start off with a couple questions just because I'm curious. So I want to ask, what particularly struck me was that the alternative pathway you talked about in pyogenes versus the pneumonia.
Okay.
Can you make any insights about where that pathway is and kind of what you expect the copper environment to be based on that pathway, or is it completely independent of kind of heavy metals going on in the cells and stuff?
So, that pathway is a really in pyogeny, it's really interesting. So it has one that kind of so it has a pathway that kind of looks like this here, but it also has a nucleotide synthesis pathway that it has like no homology. So it's really hard to make conclusions of that based off of, or make conclusions of how copper might poison that system based off of something that we have no idea what it looks like. We do see upregulation in the manganese dependent pathway, however, in pyogenes under copper stress from a microarray that we did there. It's just really hard to know. We suspect that zinc actually may be playing a role in this pyogenes as well.
Cool. You also you can stop screen sharing if you want to.
All right, let me do that.
One other question I had. So in terms of so obviously I think about environmental bacteria versus host associated bacteria, and there's probably different copper environments or heavy metal environments for things like plant associated pseudomonads versus other things.
Mm-hmm.
So do you see any like overall differences in terms of if a bacterium's entering into a host that's got copper in it? Do you see any differences there in terms of the bacterial lifestyle and the pathways that are seen, or are these pathways kind of generally present within pathogens and non-pathogens alike?
So the export system actually exists in all of them, and it's people have been using copper in like fetal pigs to actually increase growth, and we're actually starting to see duplication events in the copper export system in some of these microbes that exist in that area. But again, those aren't necessarily virulent pathogens for that environment, but who knows? Evolution, they could eventually become via gene transfer. But we don't really know what's going on in some of these other bacteria under copper stress because we just have not thought to look there. We can do the the bioinformatic thing and know that they're there, but we don't know what things are being upregulated. Who knows? What if you did take a soil pathogen or just kind of something that grows that doesn't care about invading a host, it just wants to exist. If you pushed it too hard, if it would turn something on, it's something that we want to look at, that hopefully this work will kind of, some of the things that we find out from this work can go into that.
Cool. Unrelated follow-up. Why you got a clown on Tiki Barber, man?
Hey, I said old school Tiki Barber. I didn't say he turned it around, man. He turned it. All of a sudden, he went from like fumbling it like 16 or 18 times a season to like 3. So that's why I said old school Tiki Barber, not new school. he got a lot better.
All right, I'm going to check on the different pages here. No comments there. Nothing through Twitter. All right, well, I have no more questions. If anybody else has any other questions, feel free to chime in soon. And if not, I just want to thank you again, man. This is awesome. This will also be archived later, so I'll send out the link in a few minutes to when it's archived. And thanks again, man. It's awesome, and I'll probably see you sometime soon. But.
Thanks. It was a lot of fun. All right.
Cool.